Hello everyone. Um, Thank you for coming our bi-monthly White Tulip Medical Forum speaker series. Dr. Madsen is with us today. He is very well known in the intermittent fasting era. I would like to start the introducing him, but before that, please make sure that you will mute yourself during this talk. And if you have any question, please submit in the chat box. Then we can discuss at the end of his talk. And so by that, I would like to introduce Dr. Madsen. Mark Madsen is the former chief of the Laboratory of Neuroscience at the National Institute on Aging, and is now on the faculty of neuroscience at Johns Hopkins University School of Medicine. His research has advanced an understanding of the cellular signaling mechanisms that control the formation and plasticity of neuronal networks in the brain, and cellular and molecular mechanism of brain aging and neurodegenerative disorders. His research has also elucidated how the brain responds adaptively to challenges such as fasting and exercise, and he has used that information to develop novel interventions to promote optimal brain function throughout life. Dr. Madsen is among the most highly cited neuroscientists in the world, with more than 900 publications and 180,000 citations. He was elected a fellow of the American Association for the Advancement of Science and has received many awards, including the Metropolitan Life Foundation Medical Research Award and Alzheimer's Association Zenith Award. So thank you, Dr. Madsen, for coming for this bi-monthly talk. And we are here to learn more about intermittent fasting.
Thanks for the introduction, Taha. Today, my talk, the first part of the talk, first half of the talk, will be a summary of a lot of data from animal studies and some data from human studies regarding the effects of eating patterns that include periods of time with no energy intake sufficient to cause a metabolic switch from using glucose to fats and ketones. And I'm going to talk about that in the next slide. Before I do that, I just wanted to point out one angle on this, or one fact on this that's important, and that's an evolutionary perspective. Animals in the wild, such as these wolves here surrounding this buffalo, are human ancestors prior to the agricultural revolution. Their daily routine was mainly involved in searching for food and processing food, and doing this in environments that were often harsh, that were where the food sources varied from time to time during the year, and where there's hazards and competition both within the species and between the species. And I've published one or two papers pointing out that the evolution of the human brain was largely driven by food scarcity. We know that all the early tools invented by humans were tools that served the purpose of acquiring or processing food. And there's a strong case that can be made that food scarcity has been the most important factor in evolutionary adaptation for survival in environments. Nowadays, though, both animals and pets, that is, and humans have food available too much of the time, in many cases all of the time, and they often overconsume food and pretty much put something in their mouth when they get up in the morning, several times throughout the day, and something before they go to bed. So the only time they're not taking in food is when they're sleeping. And this is, again, it's abnormal eating patterns compared to what our genetics and our evolutionary history has endowed us with in terms of responses of the cells, organs, and the organism. Okay, so typical American eating pattern, when a person eats, assuming there's some carbohydrates in the food, glucose levels in the blood go up and then they come down. You eat, glucose goes up, insulin goes up, and then the insulin helps bring the glucose back down. When they ate lunch, same thing, dinner, same thing, evening snack. And with this eating pattern, unless the person exercises, they may never switch to using fats. And the fat-derived fuel that cells use are ketones. And the next slide will go into a little more detail on what these molecules are, but essentially they're produced from triglycerides and diacylglycerols, which are broken down to fatty acids, and then they go into the liver and are converted to ketones. Both glucose and ketones can be used as energy sources for cells. Okay, and this middle example here, this person completely fasted for 1 day. So glucose levels remain low, normal levels. And the ketones remain low until late morning. And then there's this metabolic switch from glucose to ketones. The reason for this time frame here is that the liver holds about 600-700 calories of energy, and that's glucose stored in the form of glycogen. And so it takes 10, 12, 14 hours to deplete the liver glucose stores and switch to using fats and ketones. Okay, this bottom example, this person skipped breakfast, and then they ate all their food within a 6-hour time window. And so their ketones went up in the late morning until they ate. Okay, so these are the kinds of eating patterns that we refer to as intermittent fasting periods. They're eating patterns where ketones go up intermittently. It could be a couple days a week, it could be 3 days in a row, it could be every day. Okay, so with fasting and exercise, glycogen stores in the liver are depleted. The fats are broken down to free fatty acids, and they're released from the fat cells into the blood. Then those are actively moved into the liver and converted through this beta oxidation pathway to ketones. Acetoacetate and beta hydroxybutyrate are the ketones that cells can use as fuels. These ketones are actively moved from the liver to the blood and from the blood into cells. I'm showing here focus on the brain, which a lot of our work has revolved around. And in the case of the brain, there are these proteins in the membrane of neurons that are called MCTs, monocarboxylic acid transporters. And they move these ketones, and they also move lactate as well into cells. And these can be used to produce ATP. Several labs, Eric Verdin and others, had provided evidence that these ketones, in addition to being an energy source, can also have signaling functions. That is, they can affect gene expression. We found in neurons in the brain that this ketone, beta hydroxybutyrate, which increases during fasting, activates transcription factors that increase the number of mitochondria in the neurons. This is a process called mitochondrial biogenesis. And also, this ketone induces the production of a neurotrophic factor called brain-derived neurotrophic factor, or BDNF. And this BDNF plays an important role in learning and memory, in the growth of neurons, formation of new synapses, that is, communication sites between nerve cells. And also, in the 1990s, we discovered that BDNF can protect nerve cells from being damaged and killed by adverse conditions that occur in the brain during aging, and more so in Alzheimer's, Parkinson's disease. So those include free radicals, energy deficits, and other kinds of cellular stress. Whoops. All right, so this slide, it turns out if you reduce the calorie intake in rats or mice pretty severely, in this case, 40% reduction in calories below their ad libitum intake. As you can imagine, the animals maintain a lower body weight, and the sizes of different organs decrease. So liver, heart, intestines, of course, fat deposits decrease. But the one organ that doesn't decrease in size is the brain. So the brain is preferentially spared until essentially someone or an animal starves to death. Why? The brain's the most important organ in survival. This on the right shows example from an experiment where Don Ingram and his colleagues, who were colleagues of mine at National Institute on Aging, they took rats divided, when they were young, they divided them into two groups. The normal control ad libitum feeding, and every other day food deprivation or fasting. So in this group, the rats, 24 hours, they had no food. The next 24 hours, food. Next 24 hours, no food. Just keep that up until they die of essentially the diseases that kill rats and mice as they get old, cancers and kidney disease in particular. Bottom line here is the animals on this every other day intermittent fasting eating pattern lived 50% longer. So both their average lifespan and the maximum lifespan were increased. This lower graph shows the body weights. The ad libitum fed animals gained a lot of weight throughout life until they got very old. And this is mostly fat. The animals on intermittent fasting, when they're young, they still gain weight. They can gain muscle mass fine, and then they kind of level off and maintain body composition with low fat mass. So in laboratory animals, here's a list of some of the effects that have been documented in multiple studies. Increased longevity, reduction in bad fat, that's white fat. Actually, intermittent fasting will increase brown fat, which rodents have, and they use the brown fat to generate heat in cold temperatures. Increase insulin sensitivity, so improve glucose regulation. Reduce resting heart rate and blood pressure, and increased heart rate variability. This is good from cardiovascular health. Reduced inflammation, improve learning and memory, and in particular, attenuating the age-related decline in learning and memory. We found that intermittent fasting has an anti-anxiety effect once the animals are adapted to it. In animals that already have diet-induced diabetes, intermittent fasting can reverse that, normalize their glucose regulation. We found with a cardiovascular group at NIA that if we maintained rats on intermittent fasting prior to inducing a myocardial infarction, so this is coronary artery ligation and reperfusion, the amount of damage to the heart tissue was less in the animals that had been on intermittent fasting, and the function of the heart was better. And then beginning in the 1990s, in animal models of stroke, epilepsy, Alzheimer's, and Parkinson's, compared to animals fed ad libitum, the animals on intermittent fasting had less damage to nerve cells and improved functional outcomes. Now, in the case of stroke, we did a preliminary study but didn't publish it. Then another lab did a study where they induced a stroke in an animal, and this is, you occlude a vessel in the brain, and so it's like it mimics a clot forming and then resolving. And of course, in the animals, you can get pretty consistent amount of brain damage between animals. And another group found that if they wait until after they do the stroke and then switch the animals to intermittent fasting, they have improved recovery of motor function over a period of a month or so after the experimental stroke. Cancer researchers have shown decades ago that caloric restriction and intermittent fasting, with or without caloric restriction, can suppress tumor growth. And there's numerous clinical trials now of intermittent fasting in patients with cancer, particularly focused on doing the intervention when the individuals are getting chemo or radiation therapy. Because we think, based on the animal data and some early data in humans, that not only may essentially reducing energy availability to the tumor suppress its growth, but it may also protect normal cells against the damaging effects of chemo and radiation therapy. Because, as I'll show you later, one of the main general mechanisms of action at the cellular level of intermittent fasting is to engage adaptive cellular stress response pathways. That is, mechanisms in the cell that protect them against various types of stress. Now, obviously, in a heart attack or a stroke or epilepsy, these are stress on the relevant cells, and intermittent fasting is protecting. And I'll show you some of the mechanisms we think are going on in terms of gene expression and other mechanisms. Okay, just 10 seconds on this slide. Essentially, all of the risk factors for poor outcomes with COVID-19 and many other viral infections are risk factors that can be moved in a good direction by intermittent fasting. So if you are, well, in my view, not very smart and don't get a vaccine, you might consider doing intermittent fasting. Although the vaccine is going to be much better in keeping you alive. Okay, this quickly, this is a data slide measuring heart rate and blood pressure in rats before. So on the left, these two bars here are time zero. The blue bar is animals that were on every other day fasting. The black bar is animals on daily 40% calorie restriction. It turns out that these animals are also fasting about 20 hours every day, because the way these experiments are done, they get all their food at one time, and since they're getting much less than they normally get there, they eat it all right away. Anyway, heart rate goes down. This is weeks on the bottom here, so 1 week, 2 week, 3 weeks, etc. Goes down through about a month, and then stay down, both heart rate and blood pressure. But then here what we did is we switched them back to ad libitum feeding. And pretty quickly, within a week, you already see the heart rate and blood pressure going back up. And by a month, they're back to where they were. So this is kind of similar to exercise. If you quit exercising, there'll be a pretty rapid reversal of these measures here, resting heart rate and blood pressure, if you become sedentary, even if you've exercised for a long time, for years even probably. So what's happening here? Similar exercise, the intermittent fasting is enhancing activity of their parasympathetic neurons that release acetylcholine onto the heart cells and slow heart rate. And the parasympathetic nervous system also reduces blood pressure. Okay, and so you remember the sympathetic nervous system that's activated when you're under a lot of stress. It speeds up the heart rate. The parasympathetic nervous system slows it down. So that's an effect of intermittent fasting on the nervous system, in this case, the so-called autonomic nervous system. What about human studies? There's been an exponential increase in the number of controlled human studies of intermittent fasting. The most commonly used protocols are either daily time restricted eating. So the individuals there will restrict the time window they eat each day to 6 to 8 hours. So they're fasting for 16 to 18 hours every day. Another approach that we're involved in the first studies with is called 5-2 intermittent fasting, where the subject, the person will 2 days in a row each week, they'll eat only about 600 or 700 calories. And that's not enough to keep the liver glycogen stores replenished. And so the ketones are up on those 2 days. This study here, so these are among the first couple studies, and I was involved with both of them. This was in asthma patients. Jim Johnson, who's an MD down in Louisiana, he just took 12 overweight asthma patients with moderate to severe asthma. He didn't randomly send. He put all of them on a regimen where every other day they ate only 400 calories in the form of a shake. So that's very extreme. This isn't something people could maintain, long term, but these were people who were overweight or with obesity. And so this is a 2-month study. And what we found was that not right away, but between 2 weeks and a month, their asthma symptoms improved, their airflow in their lungs increased, and markers of oxidative stress and inflammation in the blood went down. And then they stayed down through 2 months. This study here with Michelle Harvey. So in this, we took 100 women at risk for breast cancer because they're overweight and have family history. Randomly assigned them to 5-2 intermittent fasting or daily calorie restriction, but eating regular meals. So the latter group ate breakfast, lunch, and dinner, but they had a 25% reduction in calories at each meal below their daily intake. And this is a 6-month study. Both groups of women lost the same amount of weight had improvements in many health indicators, markers for cardiovascular risk, glucose regulation. But the women on 5-2 intermittent fasting, even though they lost no more weight than the animals on the women that counted calories at each meal, they had greater improvement in insulin sensitivity and they lost more belly fat. And the reason is they're, on those 2 days a week, they're burning fats. Here's a more recent study in patients with insulin resistance, beneficial effects of daily time restricted eating. Here's another one looking at women who are weightlifting. And the take-home message was this, is that, and these are normal weight, healthy, young females, daily time restricted eating is conducive to maintenance and growth of your skeletal muscles. They found no effect one way or the other. The women on the intermittent fasting gained muscle mass just as well as the women who ate breakfast, lunch, and dinner. This is our review article recently. Yeah, and these are just some examples. Okay, that's my last slide before I switch to focusing on the brain. Little demographic information with probably everybody listening is aware already. The states, counties in the Southeast, there's greater incidence of obesity, type 2 diabetes, cardiovascular disease. So you're all aware of that. This is and stroke, I'm not showing here. So the obesity belt, the stroke belt, et cetera. There's a bunch of nicknames. But also, there's increased incidence of neurological disorders. Stroke is in here, so that counts for a lot, but it turns out also dementia. And of course, there's association of those poor health outcomes with physical inactivity, which is less in those regions. Childhood obesity, so you do as your parents do. You grow up in a household where people eat fast food, you're much more likely to eat fast food and stick with it. Now, there's also, if you look at academic performance, there's fewer high school graduates, so fewer of the kids with this poor metabolic health graduate high school than do kids with better metabolic health, such as my home state of Minnesota, where everybody's above average IQ and below average body weight. That's a joke, but it's actually probably true if you look at the data. And this is intelligence scores here. I would submit, so a lot of epidemiologists and so on, they focus a lot on socioeconomic status and the quality of this education. And I think it's very important to focus on the general health of these kids and their metabolic health, because that negatively impacts their brains. In fact, there's been studies done showing in adolescents, this is on average, adolescents with obesity, on average, their overall brain size is less than normal weight adolescents, and particularly in certain regions like the hippocampus, which is important for learning and memory. So I would submit, regardless of, whether these people are from poor families, if they exercise more and cut back on their energy intake, you're going to see more blue as high school graduates and more orange in IQ tests. So what's going on with the brain with intermittent fasting and also exercise? This is a simple diagram of the brain. A lot of work in my lab is focused on the hippocampus. This is a structure that lies under the cerebral cortex. It's very important in learning and memory because it's a region of the brain where all the information coming from your eyes, your ears, your nose, your taste buds, your touch receptors, your pain receptors. They initially go into the cerebral cortex, but they all funnel into the hippocampus. And learning and memories are created in the hippocampus, and short-term memories of them are stored there. And so for that reason, this has been studied a lot. It also has a relatively simple neural circuitry, which I'll show you in the next slide. Actually, all of these brain regions have this circuitry, but anyway, the most important, most abundant, and major excitatory neurotransmitter throughout the brain is glutamate, the amino acid glutamate. About upwards of 90% of the neurons in the brain deploy glutamate as their neurotransmitter. And then the second most abundant neuron uses the inhibitory neurotransmitter Gaba. So glutamate and GABA are the first and second most abundant neurons, and the most important because they're present throughout the brain. Neurons that use other neurotransmitters such as dopamine, serotonin, norepinephrine, acetylcholine, they are few in number relatively and located in discrete regions of the brain. The only way those other neurotransmitters, which I call underling neurotransmitters, affect behavior at all is by modifying the ongoing activity in these glutamatergic circuits. Okay. So here's an example of some studies we did where we're studying synapses on the dendrites. So remember, neurons have processes or extensions away from the cell body. They're called dendrites that receive information from other neurons. And then they have a long process, single process that transmits information to another neuron. And these are the axons. Okay, so in this study, what Alexis Stranahan, who was a graduate student in my lab about 14 years ago, she took normal mice, which we call wild type mice, and diabetic mice, and she randomly assigned them to 4 different conditions. In the normal laboratory condition, ad libitum feeding, sedentary, that is no running wheels in the cage, ad libitum running wheels, daily calorie restriction, which is also time restricted eating, sedentary, and then the daily energy restriction running. She let them go for 3 months. Then she euthanized them with anesthesia overdose, removed one hippocampus, and counted numbers of synapses. So this is a single dendrite of one of those glutamate deploying neurons. And these perpendicular projections, these small spine-like projections, they're called spines, are represents synapses, essentially. So what she found was that both daily energy restriction and running increased the number of synapses, and the combination of energy restriction and running resulted in the biggest increase in number of synapses. So this is the normal mice, this is the diabetic mice. The diabetic mice had fewer synapses, but nevertheless, both daily energy restriction and running significantly increased the number of synapses. The other hippocampus, Alexis measured BDNF levels, this neurotrophic factor that increases in response to fasting, exercise, and ketones. And both the energy restriction and running increased BDNF in both the normal and diabetic animals. Okay, so one effect of, in this case, daily time restricted eating and exercise is to increase number of synapses. Another effect we found is to enhance the activity of these GABAergic neurons that suppress neuronal excitability. Remember, I told you that one of the effects we saw of intermittent fasting was to protect against epileptic seizures. Some of the drugs used to treat epilepsy act by activating receptors for GABA located on these glutamatergic neurons, located on these neurons. Okay, so this is a complicated slide. I'm going to start down here where we're actually recording the relative amount of inhibition of the neurons by GABA in animals under either ad libitum feeding or intermittent fasting. And this is after the animals have been adapted from 1 month to intermittent fasting. So what you see is these downward spikes from baseline, which are more prominent here, are Gaba currents. That's essentially chloride influx through the GABA receptor channel. So we see that there's increased activity of these chloride channels. Chloride has a negative charge. When it moves into the cell, it will hyperpolarize or quiet the cell. And this may be related to the anxiolytic or anti-anxiety effects of intermittent fasting. And in fact, in the study, we tested anxiety and found that anxiety was lessened by intermittent fasting. Okay, that's all I'm going to say for that. It's a little more complicated, but I don't want to go into a lot of the molecular biology. Alzheimer's disease, it's increasing in prevalence as more people live into their 60s, 70s, 80s. We think, and actually there's good evidence that having type 2 diabetes and being overweight throughout adult life is a risk factor for age-related cognitive impairment and Alzheimer's. In Alzheimer's, if you look at the brain, there's two main abnormalities. Well, neurons degenerate, and they can be detected using a silver stain shown in black here. These are so-called neurofibrillary tangles. And then there's accumulation of amyloid. Mainly, it's most noticeable outside the cells, and it's in this yellow. So this dark yellow here is amyloid. So the amyloid plaques, and often there's degenerating neurons associated with the plaques. However, so we assume, and there's good evidence from animal studies, that neurons initially, they actually become hyperexcitable. Alzheimer's patients have increased incidence of epileptic seizures, and EEG recordings from people with mild cognitive impairment and early Alzheimer's disease document hyperexcitability. We think that hyperexcitability is contributing to the demise of the neurons. So a lot of interest in if and how the amyloid beta peptide might adversely affect neurons. Well, one finding we had a long time ago was that if we take cultured hippocampal neurons and we expose them to aggregating amyloid beta peptide, it aggregates on their surface and it impairs their ability to take up and utilize glucose. So it causes an energy deficit in the neurons. When neurons experience an energy deficit, their ability to maintain the ion gradients across the membrane and quiet themselves thereby is impaired. So we think the energy deficit in the brain in Alzheimer's is partly due to impaired glucose transport. Interestingly, however, Steve Cunan up in Canada and his colleagues, what they did is they looked at relative levels of glucose utilization and ketone utilization by brain cells in humans using a method called positron emission tomography, or PET. So they used a radiolabeled acetoacetate, ketone, and radiolabeled glucose. So they're looking at cerebral metabolic rate, acetoacetate, glucose. So they looked at this in people before and a couple weeks after switching to a ketogenic diet. So when they're not on a ketogenic diet, so glucose levels are up in the blood here, the brain cells are using pretty much exclusively glucose. But when they switch to ketogenic diet, but when they switch to a ketogenic diet, then the brain cells use the ketones and their utilization of glucose goes down relative to when they're not on the ketogenic diet. And moreover, when they plot the levels of ketones in the blood as a function of the brain cell utilization of ketones, it's a perfect linear relationship. Higher the ketones in the blood, the more ketones are used by brain cells. Why is this important? Well, Steve Cunan also has evidence that in patients with mild cognitive impairment and early Alzheimer's disease, although brain cells have difficulty using glucose, they still are able to take up ketones. And so the amyloid possibly may not have as adverse effects on the ketone transport. This right slide, the take-home message is we can, in an animal model of Alzheimer's disease, where the animals accumulate amyloid plaques and neurofibrillary tangles as they age, if we supplement their diet with a ketone ester, beta-hydroxybutyrate, it can attenuate the age-related learning and memory deficit. And it also has an anxiolytic effect, reduces their anxiety levels. This is actually data slide showing that the ketone beta-hydroxybutyrate increases BDNF in cultured neurons in a study done by postdoc Christina Morosi in my lab and by a group up at NYU who showed this in vivo. This slide just shows amyloid pathology and neurofibrillary tangle pathology in our mouse model of Alzheimer's disease. And if we start these animals on every other day fasting or daily time-restricted eating when they're very young, before there's any detectable amyloid or neurofibrillary tangle pathology, and then maintain them for a year on those diets, their learning and memory is much better than the Alzheimer's mice fed ad libitum during that time. I just have a couple slides left. Now this slide is a little complicated. It's in the New England Journal of Medicine review article that Rafa de Cabo and I wrote. I just want to look at this as kind of a, what's the take-home message? During periods of fasting or vigorous exercise, cells in the body and brain, some more than others. So obviously in exercise, muscle cells more than, and heart cells, maybe more than brain cells. Although it turns out activity of brain cells goes up in a lot of brain regions during exercise. Obviously those having to do with movement and sensory motor processing, but also cognitive. And this is a mild stress on cells. There's in excitable cells like muscle, heart, neurons, there's increased sodium, calcium influx, increased free radical production. So this is occurring during the stress. However, at the same time, transcription factors are activated that induce the expression of genes involved in, for example, removal of molecular garbage, a process called autophagy, DNA repair enzymes, genes are induced. And importantly, during these periods of bioenergetic stress, the energetic stress of exercise or fasting, the uptake of amino acids into cells is reduced. And that's reflected in this decreased activity of the so-called mTOR pathway. So the cells essentially go into conserve resources, stress resistance, don't grow yet mode during the metabolic challenge of fasting or exercise. Then during the recovery period, resting, sleeping, and eating, so eating after fasting, resting and sleeping after exercise. Then protein synthesis goes up, mTOR pathway goes up, the cells go into a growth and plasticity mode. So for example, we think that the actual number of mitochondria increases during the recovery period, but it would not increase if the genes weren't induced to set in motion the sequence of events that when the cell then takes up protein, you can get increased number of mitochondria. Okay, so the cycling back and forth between metabolic challenge and recovery may optimize health in many regards and many organisms, including humans. And then over time, there are long-term adaptations, increased insulin sensitivity, some things I haven't talked about, healthy gut microbiota. I did talk about reduced inflammation, blood pressure. You know, so these cellular events can be occurring fairly rapidly, but then there are some long-term changes that require actual growth and plasticity of cells. Okay, this is my last slide. So for people who are interested in switching their eating pattern from 3 meals a day plus snacks to daily time restricted eating, 5-2 intermittent fasting, or, you know, these aren't the only two things. I mean, you could do 2 days complete fasting every other week or something. That's better than no fasting. Kind of a key thing though is, for example, with this daily time restricted eating, it takes 2 weeks to a month for a person to adapt so that they're no longer hungry, irritable, maybe having trouble concentrating during the time period where they would have previously been eating. So for example, if you normally eat breakfast and you don't eat breakfast tomorrow, you're going to be very hungry tomorrow morning and irritable maybe. However, if you stick with it for 2 weeks, you'll find that your hunger will in the morning will diminish a lot. And by 2 weeks, it may even disappear. By a month, it very likely will disappear. And you'll find that your ability to focus, concentrate on your work in the morning is better than when you did eat breakfast. So this 2 week to 1 month period, adaptation period, is very important because people may say, well, I tried not eating breakfast, I can't do it. Well, you tried it once. If you've been sedentary and you went out and tried to run 5 miles, you can't run 5 miles. Well, duh, you're not adapted to running. So it takes time for your systems to adapt. Then what about medical education? I've, for the last, I can't remember, 6-8 years, I've taught a course to the Hopkins medical students. Not a course, sorry. An hour and a half lecture to them on intermittent fasting. And there's a number of clinicians at Hopkins that have done, are in the process of performing or trying to get money to perform trials of intermittent fasting in patients ranging from those with multiple sclerosis or cancer to, well, obesity, which is what it's been best demonstrated to help with. Epilepsy is another one in combination with ketogenic diet, for example. Now, of course, there's no financial incentive to do this, but I would hope physicians will learn that for someone to change their eating pattern or initiate an exercise program, they need help. They need cheerleading. And you can't just have them come in and you tell them, oh, you're overweight and your blood glucose is getting a little high, you need to exercise and cut back on calorie intake. They turn them loose for a year and they come back. There's got to be some way the system changes so that people can be helped to get through 2 weeks to a month, so at least they can have a legitimate chance of adopting these healthy type lifestyles. These are just some examples of ongoing clinical trials of intermittent fasting in various diseases. So many of which, and this is, well, this is quite a few years ago. This is 8 years ago, my lab at NIA. I retired in 2019, and this is a bunch of postdocs, graduate students, and collaborators over the years who gave me a symposium for my retirement. So thank you very much.
Thank you, Dr. Madsen. This is a great talk. I mean, each one of your slides can be used to implement, to mitigate different types of risk factors nationwide as well as in the world. So we have a couple questions. If you don't mind, can I go over those questions?
Yeah.
And get answered? So one of the person asked, did anyone compare the aspirin and aspirin plus intermittent fasting for stroke prevention or other neurological disorders as an add-on lifestyle modification to mainstream treatment?
No. Now so the aspirin story, my understanding is like with cardiovascular disease, recently there are some studies saying it's not as clear as it was initially thought to be, low dose aspirin being beneficial. I guess the stroke data there hasn't been any change in thinking there. But no, there have been no trials of combination of intermittent fasting with asthma, with aspirin.
Sure.
Yeah.
Okay. Yeah. So the other question is there any study about intermittent fasting effect on patients have eating disorders such as anorexia, nervosa, or bulimia? If there is not any, what do you think about intermittent fasting effect on eating disorder?
It's a very good question. Intuitively, this kind of gut feeling is that someone with an eating disorder might not be a good idea. But there are no data, so we don't know for sure. This is psychiatric disorder, kind of an obsessive compulsive type psychiatric disorder, and it's usually young, adolescent girls, and they tend to not only restrict calories but exercise a lot. Some of this is thought to have to do with body image and so on. But yeah, I wouldn't advise that as something they should try. It just doesn't seem like a good idea. Now, having said that, from an evolutionary perspective, any species would die off if individuals starve themselves to death. So it's an unusual psychiatric disorder that, to my knowledge anyway, is peculiar to humans. And so that's all I can say about that.
Okay, so the other question was, what about the dry fasting on the health benefit? Has anyone looked up the dry fasting compared to the, I guess, wet fasting?
Well, I think they're saying fasting with not taking liquid either.
Right.
Now, I think it's important to stay hydrated. That's just, but I'm not aware of any, even animal studies, where they've combined, food and water deprivation. Of course, we don't live long without water. Rats and mice even die sooner without water. So, boy, I don't think it's a good idea to do water restriction.
But there's no data, right? There's no science.
No.
Okay. So the other question is, as a result of your studies, which one is more effective for both health benefits and weight loss? 2 to 5 or time-limited eating?
They've never been compared directly. All the studies that have been done, to my knowledge, and published, it was always comparing 3 meals a day to either daily time restricted eating or 5-2, but not all of them in the same study. So we don't know. All we know is compared to 3 meals a day plus snacks, either one is better.
Okay, great. So that's pretty much it from the question session as well. Thank you, Dr. Madsen. This is very helpful. I remember Dr. Stanley Hazen's talk when you were talking about the fasting and the longevity benefits. So this is awesome. Thank you for everyone's attendance. We look forward to see you in a monthly speaker series. Thank you, Dr. Madsen, again.
Okay, goodbye.
Bye-bye.